So yeah, welcome to this slightly altered date for the Space Force. We didn't have a meeting on July the 5th because of the holiday, so we pushed it to today, obviously. And today we're really excited to have Joe Beecham talk from NanoString Technologies in Seattle. So Joe got his a Ph.D. in biophysics from Johns Hopkins, which I think explains the kind of technological trajectory of his career. And Joe is now the senior VP of Research and Development at NanoString, and is the co-inventor of the NanoString GeoMx digital spatial profiler, which I know many of us in San Diego are familiar with, but also the co-inventor of the next generation instrument, which is the Spatial Molecular Imager platform, which is going to be launched next year in 2022, which has more of a single cell and subcellular capability. So I think it goes without saying that many of us are very excited in San Diego about these types of technologies. So it's really great to have Joe to come and speak to us a bit more about NanoString's contributions in these areas. And I think it's clear, definitely the case that NanoString are a leader in these technologies. So thank you very much for speaking to us, Joe.
Hey, thanks, Peter. It's great to be here at this series, and let me see if I can share my screen here and get this thing started. Okay. Now I have to get it in the right window. Is that the window? Is that the right window I'm in?
It says launch meeting. We've got a launch a Zoom meeting window. We had your slide.
Okay, got it. Okay, I'll come back over here. I never know which
that one's right.
Monitor this thing's going to come up in. Let me get the duplicate slide thing going. Okay, can you see that, Peter?
Yeah, that's great.
Okay, great. So, hey, it's great to be talking to Sanford Burnham. It's been a while since I've been down there. Actually, I think I was looking at the last talk I did down there was probably about, I think it's like 4 years ago. But I always like being down there, and it's too bad I'm not down with you guys today. But I'm going to describe to you today the two spatial biology platforms we're working on with just a little bit of introduction. I think a whole lot of us lived through a lot of these technologies being developed back when Next Gen Sequencing won Method of the Year back in 2009, all the way to where a high resolution spatial biology won it here in 2020. And we've really progressed. We went to the place where we felt that we could take tissues and bulk them, basically blend them up, and then just sequence away. We learned a lot. TCGA was a great database that came out of that. But the importance of rare cells for a lot of things, especially in cancer, really, we required new technologies to look at rare cells. Single cell RNA-seq really came to the forefront, Method of the Year in 2013. It's a great technology. And, you end up with a parts list, which is great because you can see rare cells in a parts list. But it's the most unsatisfying part of that whole thing is that you don't know how those parts work together in an organized structure to make functions. So I was sort of back here in early 2013 and 14, really frustrated by the fact that I wanted to put the parts lists together in an organized spatial manner and try to figure out how they worked. And that's sort of the origin of how the digital spatial profiler was born. And I'll tell you a little bit about that. But I do think we're entering an incredibly exciting era for spatial biology where we are going to go from discovery where you end up with abstract cell types in a UMAP to where we actually take all those cells in a UMAP and put them into spatial context. We got an X, Y, and Z coordinate for everything in a UMAP. And I'll show you what that data looks like. We're going to go from places where it may take 20 slides to do 80 proteins. Now you do 100 proteins on one slide, pretty straightforward. And then, you know, the final translational application of this is we're really going to be able to improve the information content of our biomarkers for both diagnostics, but drug development, mechanism of action. So that's sort of the promise of where spatial biology is going to take us. And then let me talk to you about the two platforms we developed. And I think it's important to think about spatial biology. I really do think of it happening over multiple spatial domains. And if you actually look at sort of the physiologically relevant unit in a tissue in terms of a function, what you find is that those spatial domains range from about 100 microns out to multiple millimeters. So like a glomerulus of a kidney is about 100 microns. There's 2 million of those in a kidney, but they really don't function as single cells. They're organized groups of cells. And we really developed the GeoMx to actually adapt to the spatial shape of the unique tissue geometry that exists, and then do multi-omic, either whole transcriptomes or hundreds of proteins at a time along those unique shapes. And I'll describe how that works. Now, at the same time, these organized structures actually are formed from single cells that actually have to differentiate and self-organize into those particular structures. And to study that mechanism, that spatial axis, we developed the spatial molecular imager, which allows you to go subcellular and then actually understand really the private life of cells and how they get organized. And I'll describe both of these technologies today. Now, it was just about 5.5 years ago, almost exactly, where I was giving a talk down at MD Anderson, and actually during work for, through this COVID thing, I got a question from somebody who was listening to one of my talks in India and said, could you come up with the original idea? Could you find the original idea? And I remembered where I was in the lobby of the MD, out in front of MD Anderson. I always stay at a Marriott there. I was going to give a talk, and I scribbled this thing down that you see in the center of the screen here, which is scribbled down as an idea on how to change to do unlimited plex in a multi-omic fashion in FFP tissue. And the whole concept was based around using light as your spatial resolution axis, but then having your affinity reagents connected to sequencing barcodes through a photocleavable linker. And that concept allows you to do all sorts of, I think, pretty interesting HighPlex profiling. And after that talk, I actually sat in the office of a guy there, Gordon Mills, and he was a protein expert, and we sort of sketched out the fundamental makings of the GeoMx I'm going to describe to you today. And it pretty much worked right out of this memo pad that was scribbled on in a lobby of a Marriott in front of MD Anderson, on whatever that was, about 5.5 years ago. And I'm also going to tell you about another technology we developed, spatial molecular imager, which actually came from a totally different area. We were inventing and developing small 20 nanometer sized optical barcodes that would allow you to sequence windows of DNA or RNA. And it was called a hide and seek technology. We actually developed it. You can see we started working on those optical barcodes back in 2016. A semiconductor company came in, actually gave us $50 million just to watch us do that. Because they wanted to learn how to do sort of new types of technologies in life sciences. So Lam Research came on. We had a lot of money to develop this chemistry. And then the killer application of that chemistry, it was a hybridization-based way of reading nucleic acids. And then back in June of 2019, I decided to take the entire program and actually move it to become an in situ imager. And that's the spatial molecular imager that I'll describe to you today also. So that's sort of the backgrounds of where those two chemistries came from. Let's talk about the GeoMx. One of the key aspects of the GeoMx is that, again, you can see the affinity reagents, whether you're doing protein detection with antibodies or you're doing in situ hybridization with fish probes, is that we do our multiplexing with a 60 base pair piece of DNA. So that has the UMI, it has a 12-base indicator that tells me what analyte I'm connected to, and it's got handles for PCR and things like that. But it's connected to the affinity reagent to a photocleavable linker. Same exact structure, actually, the same sequencing structure is on the back of our fish probes, but they're all connected through these photocleavable linkers. And what that allows you to do, it's really important, is that every tissue's geometry is unique. If you look at enough of them, and I have, no two tissue has the same geometry. And the kind of biology you want to measure is uniquely linked to the way that tissue is constructed. And it's not constructed on an XY grid. You can't probe tissue with an XY grid and really get what you want. But you can if you have adaptive mirrors. So we put in the back of the GeoMx 2 semiconductor chips at around 1 million micrometers. Each mirror is 1 square micron in the image plane of the tissue. So it's 1 square micron, and it will adapt to the tissue geometry itself and allow you to do HighPlex profiling with a shape factor that conforms to the geometry of the tissue. So this is the GeoMx digital spatial profiler. You see the box over on your left. And it works just like you would expect. All the chemistries we developed were all developed with FFP tissue. It works on fresh frozen, fixed frozen. But from the very beginning, all of our chemistries work with HighPlex, Multi-Omic on standard 5 micron thick or whatever thickness you typically work with, biobanked FFP samples. So you start out with one of those slides. You put your affinity reagents on, and if you're in the RNA world, could be 22,000 fish probes you're doing at a time. Or if you're in the protein world, it's typically 100 to 150 antibodies at a time. They distribute with respect to the way the proteins and the RNA are distributed by Mother Nature, if you will, on that tissue slice. They're invisible, so you don't see anything. So we dope in 4 colors of fluorescence and we scan the entire tissue. So you can see the overall geometry of the tissue at a 4-color level. Find unique, interesting regions like where the immune infiltrates are or where 2 different tissue boundaries come together. You go to that region of the tissue, you turn off all the fluorescence, you shine a brief burst of UV light that will liberate the sequencing barcodes from your affinity reagents. Again, it's all in situ. It's a non-destructive way of probing the tissue. The sequencing barcodes come off in step 4. You literally drink the photocleaved barcodes off the affinity reagents with a small microcapillary, and you put it into an on-instrument microtiter plate. This entire operation takes about 45 seconds. So you wander through the tissue, generating what I call digital replicas of the affinity reagents in space. You fill up your microtiter plate, you go, it's just unlimited how many, we call those areas of illumination you want to collect. And then if you're underneath 800 plex or so, you can just read them out on a normal N-counter readout. When you want to go all the way up to 22,000 plex, then you read them out on a next generation sequencer. This gives you an idea of the kind of content that we have. We're now over 300 validated barcoded antibodies that work in the area of immuno-oncology primarily and in neurosciences. And we continue to expand that every year by a couple of hundred antibodies. And then over here, we have our next-gen sequencing readout. We actually started with 2,000 genes. And then over the last couple of years, we've advanced it all the way up to 22,000 genes in space that you can probe either human or mouse tissues with. And then I'll describe a little bit, but the unique character, this is not HighPlex profiling over a grid. This is HighPlex profiling over the shape unique to each of these tissues. So this gives you an idea. This is a colorectal cancer sample we're looking at here. And one of the 4 colors of fluorescence we put in there is a pan-cytokeratin antibody that's got a yellow fluorescence dye on it. And you can see that lights up all the tumor-specific cells in this particular sample. We also have an antibody labeled in magenta here that will light up CD45, which is the immune microenvironment of that particular cancer sample. Now, the Geomix will take a picture of this, and then it will actually align all the micrometers with single cell resolution to mimic the exact geometry of the tumor-specific cells that's got the Alexafluor 594 dye on it. And then that becomes your UV mask for shining the UV light onto that particular cell, onto that particular sample. And it will liberate, say, the whole transcriptome from just the tumor compartment of that particular mixture of cells. It's about half tumor microenvironment here. Then the mirrors can actually invert or actually image off the CD45. You do another 30-second operation where you shine the light, and then the whole transcriptome that's unique to just that tumor microenvironment comes out, and that will be 2 positions of the microtiter plate that you're looking at. And you just wander through the entire sample like this, collecting tumor and tumor microenvironment whole transcriptomes or HighPlex protein. When you do that, there's a lot of work now out on the HighPlex protein. So I'm gonna sort of short shrift that one a little bit, but because a lot of stuff is in the literature. This is what non-small cell lung cancer looks like in the Geomix. We're looking at various compartments here. You can see the tumor compartment in green, CD45 in yellow, sometimes CD68 in blue, and CD45 and then stromal. These are non-immune containing stromal cells. But all those arrangements would be mirrors would adapt to those particular geometries. And we do HighPlex protein profiling typically. This is work done by David Rim up at Yale Pathology. He had a whole cohort that was in TMAs. They were all treated with checkpoint inhibitors. And then they used this HighPlex profiling to determine predictive biomarkers for this particular type of checkpoint therapy. We now have about 60 papers of this type out there in protein, so I won't go into them in detail. Just mention like our very first 2 papers actually on the Geomix were back-to-back papers in Nature Medicine. And actually the winner of the Nobel Prize for Immuno-Oncology, Jim Allison, was actually a co-author on the very first Geomix DSP paper that came out that was done back-to-back there. A number of papers come out. We've been on the cover of Clinical Cancer Research, a bunch of other areas. But we actually have quite an array of papers now that are doing HighPlex, predominantly protein work in these various areas. We actually track these things. It's kind of nice because if you're trying to get peer-reviewed papers accepted, we now have 4 papers accepted inside the Nature journal. You can see what I got 4 in Nature Medicine. I think it's actually 6 now. But getting raw data at this level, peer-reviewed and published, it goes, as you guys know, you really have to have really robust, reproducible data that the outside world can find orthogonal, and we did orthogonal verifications and validations on a lot of these things. So it's been very successful. Now, we weren't stopping with just developing the protein layer of information in Geomix. Actually, when I gave a talk at AGBT in 2019, actually, when we came out with 84 genes in space, it was actually about a factor of almost 10 greater than any other spatially resolved RNA at that time from a commercial source anyway. And that was 2019. And over the last 2 years, the talk I gave at AGBT in March of this year, 2021, we're talking about and showed data and now commercially released 22,000 genes in space. So we've gone from 84 to 22,000 in just 2 years of development. Nice thing is that RNA scales like this. And once you're at the whole transcriptome, it basically opens up all the biology available in neuro, cardiovascular, developmental. It really doesn't matter. You can actually probe all sort of life science biology with these kinds of reagents now. And let me just show you a little bit of what that looks like. We actually formed a partnership with Illumina early on. We wanted to generate what we call sort of push button whole transcriptomes in space. We interacted once Gary Schroth, who's sort of like my counterpart at Illumina, sort of like a CSO there. once he saw the quality of our spatial data coming out, readout on the Illumina, we actually wanted to try to make push button whole transcriptome. So we interacted with them. We interacted with the lead scientist in Dragon and Edico when they came over. And we sort of implemented sort of almost push button whole transcriptomes on FFP. And then let me tell you a little bit of a surprise on some of the data that comes out. And I'm sort of picking something here that's a little bit different. You take some area like a kidney, you just look at a normal kidney. And then you can just take it and start profiling the key histological functional domains of a kidney. You can see a glomerulus there. That's where you actually start making urine, and a whole bunch of other things happen in here with the high blood pressure and a lot of other things. That material goes into what's called a proximal convoluted tubule, goes down through a thing called a loop of Henle into a distal convoluted tubule, and then out. And we can just go in and actually take and generate whole transcriptomes on these, what I call histologically functional regions of a kidney, and say, what's the information content of the whole transcriptome? So this is what it looks like when you do that. There's your kidney. Here's the DSP image that we're looking at. There's the cortex of a kidney. There's the medulla, it's sort of more in green. The mirrors in the GeoMx can actually adopt to the exact shapes of these unique physiological features, physiological features of the kidney. You can see them there on here. And then now we're generating whole transcriptomes as I walk through sort of the spatial axis of a kidney. Now, what's sort of amazing to me is actually what you find is that we basically recapitulated about 50 years of kidney physiology in a single experiment. About 50 years of research determined how all the transporters change as you move from a glomerulus out through these different regions. And if you just do whole transcriptomes, this is a simple 5 micron FFP kidney sample here. And then I just map all the transporter pathways I see when I'm in the proximal convoluted tubule, say. Those things all map to what kidney physiologists have found, like the sodium glucose co-transporters up there. Yeah, it shows up exactly when I'm in that region of the proximal convoluted tubules. And as you move down physiologically, if you will, sort of the loop of Henle and the distal collecting duct, what do you see? You see a whole bunch of different transporters show up, metal ion transporter, choline, and bicarbonate. Yeah, lo and behold, that's exactly what the physiological world determined shows up there. You can continue working your way down. Actually, I don't show that. When you get down to the final collecting duct, it's the ammonium transporters that light up, and you can see that down here. But you can see how you're recapitulating like 50 years of electrophysiology by just plotting the whole transcriptome transporter pathways as you move through space. Now you can say to yourself, how do we know it's exactly real? And we got a bunch of things we did with that. But one of the ones that surprised me was this, is that you can't actually, I can't go to a database and say, okay, what's a whole transcriptome of a glomerulus look like? It doesn't exist anywhere. But what you can do is you can go to the Human Protein Atlas, look in that database and say, what proteins did they find in the glomerulus, say, of a kidney? And you can do that for all these different substructures. It doesn't matter what organs. And when we do that, we find that I can plot Human Protein Atlas protein location, subcellular and histological structure location against the GeoMx whole transcriptome, and they just map amazingly well. It's hard to see it when you're looking at thousands of these guys on one graph, but I'll pick a couple of them out so you can get a feeling for how this works. This is an area I'm in the distal, okay, I am in the distal convoluted tubule. So this is an HPA image of a protein called calbindin 1. And you can see where it shows up. And that looks like just exactly my mirror configuration for distal convoluted tubules, because that's a histological feature that my mirrors can align with. And then on the right-hand side, out of whatever, 19,000 transcripts I'm seeing in the whole transcriptome, nothing in the collecting ducts. I hit the distal tubules and lo and behold, that's exactly where you see calbindin 1 mRNA show up. And I go over into glomerulus, nothing, filtration membrane, nothing, loop Henry, so on and so forth. And you can see how, one transcript out of the whole transcriptome maps exactly where the HPA found that particular protein. And there's a ton of them like this. Here's one solute carrier family 34, whatever that is. It doesn't show up anywhere in the other parts of the tube of the kidney. I hit the proximal tubule, which is where HPA found it. And that's exactly where that transcript shows up. So that's a great example of a sort of a totally orthogonal way of validating some of these studies like this. Okay, now when you look at a sample, that's just like pure discovery work we're doing there. But if you're looking at a cancer sample, this would be an excisional biopsy. This is actually colorectal cancer we're looking at here. And we're saying it's a 5 micron. And we're going to do whole transcriptome with all these different regions. So I'm doing about 100 whole transcriptomes in this particular region of this colorectal sample. It's 300 micron regions, but each one of those 300 micron regions are cut up into tumor microenvironment versus tumor. Then you ask yourself, how many genes do I measure on average out of the whole transcriptome in these 2 compartmentalized segments? And the answer is about 9,000 genes in general you'll measure along these unique geometries as you go through a typical excisional biopsy in cancer. And then what you find is that it's amazing how these whole transcriptomes change in space as you move through these excisional biopsy samples. So one of the ways to visualize that is we set it up you make a Circos plot, you put the whole transcriptome like hourglass or hours of your clock. And we're going to put both the tumor whole transcriptomes around the hours of the clock in green, as well as the tumor microenvironment and sort of orange around there. And then you can just watch these transcriptomes change as you move around. Now we organize the pathways. You can see cytokine signaling, extracellular matrix up here between 12 and 1. And you can organize these things this way. And then just watch it, how they change. And I'm looking, say, at 2 regions over here to maybe differ by a couple tenths of a millimeter, say. And you get over here and the cytokine, chemokine signaling pathways are all hot. And I move 2 millimeters to the left. And then this is totally not hot region of that tumor slice. Cell cycles are going up and down. Cell-cell interactions are going up and down as I move through the geometry of that tissue. We actually did a bunch of studies on this of this type. You can follow things like T-cell exhaustion through IDO1 pathway mapping. And a lot of this work is actually all in press now, so I'm not going to go over too much in detail, but it's sort of the way whole transcriptomes in a particular application area like cancer shows up. Now, when you're doing something like in mouse, you're doing 22,000 genes in a mouse. Again, those transcriptomes can be shaped exactly like the mouse tissue is shaped. So when you go into a brain, you're not just doing a grid pattern with whole transcriptomes. You're doing things like there where the whole transcriptomes map with a cell layer like this. And you get these unique geometric patterns that are all linked to the way a mouse brain is organized. And then you end up with all your mouse whole transcriptomes following really the way the cells are organized in a mouse brain. And we're doing a bunch of studies of that type now with 22,000 genes at a time. So that's sort of what I can tell you, a brief introduction to how GeoMx digital spatial profiling works. And let me tell you a little bit about how that multicellular sort of unique whole transcriptomes and HighPlex proteins that are linked to histological features, physiological features of a tissue get translated down to when you go subcellular and you look at how single cells are organized. And that's why we developed the spatial molecular imager. It's what some of the data of the SMI looks like. I sort of call this thing the Hubble Telescope of spatial biology, because we're typically doing on the order of about 500,000 cells and 1 billion transcripts per sample on this particular device. this is what 1,013 plex of RNAs look like. And I'll describe a little bit how that works. You actually can't with your eyes look at 1,013 different colors here, but let me tell you how we get information out of these images. I'll blow them up a little bit so you get a better view. Each one of those dots is a single RNA molecule that has one of our little 20 nanometer size barcodes hybridized to it. And that barcode is going to change color, and that color pattern represents the multiplexing that we're doing in these samples. And again, we developed this chemistry initially to actually build a clinical sequencer for FFP that was all hybridization based. And this is what it looks like on the left-hand side of the screen when you take an FFP slice, and then I'd be extracting the RNA out of it, pulling it down onto an ordered array chip, and that's why you can see these little patterns. And then taking the little 20 nanometer barcodes, and they would all light up in different colors, and we would sequence windows of that nucleic acid. When you think about that, it's like it's all hybridization based. Doesn't it seem crazy to some extent to take Mother Nature's ordered array that actually has some physiological function associated with it? You know, why do we take it out just to put it down onto something that's an artificial ordered array? So we decided to leave Mother Nature's RNA and proteins alone and take our hybridization barcode and just go in situ and see if we can make this work. So we started that a couple of years ago, and this is where we're at on the thing now. Again, it works with normal biobanked FFP samples or fresh frozen or fixed frozen if you have that, but it works with the billions and billions of these FFP samples that are out there. It's a normal IHC style front end, either IHC or in situ hybridization kind of front end. So nearly identical to GeoMx. In fact, these are all automatable on a Lycobod tissue processor. Like GeoMx is already automated that way, but SMI will be done the same way. That's what the SMI looks like. It has 4 cells that actually you're imaging at a time for samples. These are the bulk reagents. There's a unique cartridge for the unique 20 nanometer barcode reagents coming over here. And then this is the waste. A single integrated unit that actually gives you all this information. You get your UMAP plots. Each cluster, each cell over here in a UMAP's got an X, Y, and Z coordinates, so you can put it back together. And then you end up with gene expression along all these different spatial axes. I won't describe in detail the chemistry. It is a cyclic chemistry. I think I have one blow up image of this, but again, here it works a lot like GeoMx in some respects. Here's Mother Nature's RNAs that are in situ. Here's the business end of the hybridization. Here's the little tail. And instead of having a photocleavable tail here like we do in GeoMx, we have a piece of nucleic acid that we actually read through cycling of the reagents. Our little 20 nanometers will change color with a pattern that tells me what RNA I'm connected to. Again, in situ, it's non-destructive. You never try to grab the RNA and pull it out. And the color patterns change. Currently, we're working with 64 bits of information encoded in that 20 nanometer barcode, which allows you to do nearly unlimited plexing. I'm just going to be showing you data today at 1,013 plex. And you end up, here's a blow up of a region. There's one transcript sitting here. And in this particular transcript, as you cycle, it goes from blue to an orange yellow, back to blue. And those are all encoded information that tells me what particular transcript is located exactly at that X, Y, and Z coordinate axis. That's how it works. The data density is really pretty remarkable. The raw data is about 10 terabytes of data. We never pull it out of the instrument. We never move it. That 10 terabytes get crunched down to about a terabyte of fully digital information. And then we wrap additional information around that, like where does one cell end and another cell start? And we end up with a completely digital batch of 3-dimensional data that's about a terabyte per sample examined. Let me tell you a little bit about the 1,013 plex panel we're looking at. If you look at how much plex do you need to really understand how 2 cells talk to each other, what type they are, and how they interact. It turns out you need about 1,000 genes to do a half decent job of that. So we got about 243 genes devoted to cell typing. We have about 270 genes devoted to cell state and function. We got about 435 genes devoted to how two cells talk to each other. About 46 genes for some remote hormonal activity, which does happen in these tissue slices, and they're all put together at subcellular resolution, and they're all multiplexed at one time. This is what it looks like when you're in different types of biobanked FFP tissues. Don't matter, melanomas, non-small cell lung, renal cell carcinomas, colorectal, tonsil. Each one of those little dots would be a uniquely cell ID. We'd be using those 250 genes. We actually use all 1,013, but 250 of those genes are specifically designed to ask their cell types. And you can typically monitor up to 30 or so cell types in this manner. Then you have all those other genes to determine what are other kinds of information. You can actually cut little tissue slices up, and you can see here in a 6 by 5 millimeter space, I'm looking at about whatever it is, 4, 8, 10, 12, 15 different samples at a time we're looking at there. And actually we had this with a goal in mind. We got heat maps of all the cell types we expected to exist in these different tissue slices. So we were able to recapitulate that. We also have made a ton of use out of the CCLE database that the Broad put together, which are about 1,013 different cell lines. These are mainly cancer, solid, and liquid tumor cell lines. And we make sure all of our whole transcriptome stuff maps and correlates with the CCLE. We do the same thing here with our SMI. We make FFP cell pellets of all these different cell lines and generate correlation graphs with deep RNA-seq sequencing of maybe 500,000 cells. And we do this at the single cell level, and you get nice correlations around that just tells you that your 1,013 plex is following the biology that exists inside the CCLE database. And then you go in and you do biology in real tissues with that. Now, one of the things that you find is important here is that the protein layer is important. RNA is great, but it actually doesn't really give you much about the tissue morphology and how the structure of a tissue is put together. So we actually simultaneously do RNA work. And you can see here, RNA is much better at actually determining cell boundaries. This is a blow up. So we're actually determining real cell boundaries here. There's little white lines around each of these cells. The DAPI nucleus is in there, and we image the DAPI nucleus, but I don't try to extrapolate where the cell stops or anything from the DAPI. It's just another piece of information that we include in all of our HighPlex protein work that determines the overall structure of the thing that we're measuring HighPlex RNA in. And we find it really useful to have a data-driven segmentation, if you will. When you look at how this thing works, to give you an idea, we're measuring about 1,250 transcripts on average per cell with our 1,013 plex. We oftentimes have as many as 4,500 transcripts that we are locating with a little 20 nanometer barcode in 3 dimensions there. If you're looking at the background signals, we include ERCC alien control sequences that go through the whole thing. They got their own barcodes, and it's a great way to get negative controls in situ. The noise on our in situ is about 1/10 of a transcript per cell. So if you're looking at something with 1 or 2 copies per cell at the RNA level, you're almost a full log unit above the noise level of the spatial molecular imager. And then, of course, when you do real experiments, this is a renal cell carcinoma we're looking at here. You generate an image. Here's the UMAP plot of the various cell clusterings. Now, every one of those cells here now has an X, Y, and Z coordinate that we can put them back in space. And that's what you see over here. We never took them out of space. It's just one way to represent the data. And then we got about 1,000 genes to do sort of spatially resolved changes in phenotype of these cells. And then you do see, even within these cell classes, you get unique functional gene expression just where you expect. It's where 2 boundaries of different cell types come together. It's actually tumor on this side, immune over here. You get a unique layer of gene expression that actually maps where those two boundaries are. And we see that all the time in geomics, right? The action is right where these boundaries of cells come together. And then you can superpose these things. On the bottom right, we got unique rare T cells that we're looking at here with this particular investigator, and also monitoring how those T cells change as they go into different spatial locations of the tissue. We had a poster on this at AGBT, and this is work we do with the discoverer of bystander T cells in tissues, a guy named Evan Newell at the Hutch. This is what the data looks like in 3 dimensions when it comes out. Got a little movie. That little glowing area is a single DAPI nucleus of a single cell. So we're looking at what, about 5 or 6 cells here. The little transcripts come out in X, Y, and Z coordinates. You can see them coming out in the plane and above and below the plane there. All this information is gathered at this level. It turned out, in this particular panel, I had various non-coding RNAs in here. Some of them map uniquely to the nucleus. And one of them is called mallet1. It's a little orange transcript that's in here. I think I take away a whole bunch of colors and give you an idea. You can see how mallet1, the orange ones, in 3 dimensions, they all map with the nucleus. I'm a little short on time, so I'm not going to be able to show you the whole movie. But anybody has an interest, I can bring that one back up again. And then let me end on just one aspect of the SMI. You take this technology, same way we're imaging with a 20 nanometer barcode RNAs, we're going to do the same thing at the protein level. Except now, proteins don't look like little punctate spots. They do when they're lowly expressed, but in general, you see different kinds of patterns. This gives you an idea when you cycle on protein targets, and your eyes are great at seeing 2 things at a time. So this is a case where I got a barcode on there, and CD45, it shows up green in this particular cycle. Histone H3 shows up red, so you can see a nucleus versus the cell membrane there. Then I cycle the barcodes on to cycle 2, and the rules on our CD45 antibodies is bind to a red 20 nanometer little barcode this time, not the green as they cycle through, and then histone change. So you can actually see with your eyes sort of the encoding scheme on a simple 2-plex. Now, when you do it for real, you're working on encoding schemes that are bigger than that. So you end up with these images that are combinations of colors, all depending upon how you engineered and how you encoded information in your little barcode. You can see some things like these are negative controls, so they hardly show up. But like on cycle, let's pick one out. Oh, cycle 6, say, these 3 targets would all be looking blue. One of them would be green. So that's when you look at a spot 6 image, it almost looks like a completely blue image there in various combinations. Of course, your brain can't encode all that information, but it's simple to take those patterns and get the molecular targets over here shown to your right. But it really is different. I'm just sort of emphasizing how different these images at the protein layer look like compared to the RNA, which is all a bunch of punctate dots. And now we're putting those 2 things together into one experiment where the RNA layer gives you this beautiful structural information on the tissue architecture. And then the RNA gives you a wide swath of biology that happens together. And you put both those things together to generate a complete experiment on SMI. So this is my last slide there, Peter. So we're going to leave about 15 minutes worth of Q&A, but just in the end, let me emphasize, we got these two spatial biology platforms. They've all been designed to be the unlimited plex. So we're at unlimited plex over here at GeoMx, whole transcriptome. We're doing large panels. These are FFP, the largest kinds of panels that are currently be done with FFP right now. They work with our multi-omic. So all of these technologies work with both protein and RNA layers at high sensitivity. And you're collecting over 9,000 genes when you're doing whole transcriptomes and these unique geometries. All of them are multi-omic, RNA plus protein. And the Genomics is designed, it really is designed to follow the tissue geometry. And there's just a million examples of that. I didn't have time to really go into a lot of the interesting cancer examples. There's one down here where you actually make little 50 micron expressway lanes of how the immune cells interact with a border of a tumor, say. That's where you're adapting to the tissue structure. And then again, these are all totally automated systems so that data collected in one place can be reproduced in another place. So I think that's a good place to stop, Peter, and maybe I'll stop sharing and maybe we can get some questions going.
Yeah, that's excellent, Joe. Thanks very much. It's really good to hear a bit more about the SMI. So there's a few questions coming through. I'll just, to everybody, if you could put questions in the chat and I will read them out. Jeff, Jeff, you should be able to unmute and ask questions yourself because you're a panelist. Same for Brian, but if not, just type them in the chat and I'll read them out, and the same goes for everybody else. So Joe, one question that I'm still not sure. I should probably know the answer to this, but I haven't quite figured it out. So you emphasized how that with the DSP, the regions of interest can kind of follow the the histological structure of the tissue.
Yes.
So that means then that regions of interest can vary in size quite a lot. So does that have any impact on the quality of the data?
Yeah. So when
comes out of
right.
it.
Yeah, you may be in one part of the tumor and your T cells are coming in, and then there's like maybe there's whatever. We count every cell that the mirror images. So let's say you had 102 tumor cells there, you're doing whole transcriptome, and then on the immune side, you may have done 53 T cells, say. Yeah, we record all that information. So when you get your whole transcriptomes out, you'll know that came from 103 cells and the other one came from whatever, 53 cells. So you can actually normalize that on a per cell basis if you want. We also record the exact area. Some cells have more cytoplasm than others. We record the exact square microns
right
there.
But does the number of cells affect the quality of the data? I mean, I know there's a lower limit. You need a certain number of cells to be able to.
Again, remember, these are all digital counts. These are all digital counts. It's not like the data gets noisy. It doesn't get noisy. Each one of them is a single molecule counted barcode. What you'll find though is you go down, if you're looking at the range of transcripts in terms of, say, expression levels, say from 1 copy per cell to whatever, hundreds or thousands of copies per cell. When you go and you get down to the very small ROI regions, and some of the data that's coming out in the brain goes down to about 4 cells. That 4 cells in a brain, you see about 2,000 transcripts, to give you an idea. They don't get noisy. What happens is the low end will drop off. So you will not see, there'll be certain genes you'll just not see. But the data you get on all the ones you do, just like in a single cell RNA-seq experiment, is high quality data, the ones you observe.
Okay, but for larger regions, then you get more genes.
Sure. Yes, you will.
Right.
Yeah.
Okay.
And actually, it's the presence. It's not like a linear function. You actually get to sort of maximal genes really pretty quickly in space in terms of that.
Okay.
What you find is that then what you're doing after that is the dynamic range in which you measure that gene. Sort of people sometimes forget that there's a dynamic range term. And it's really important, actually, if you're trying to quantitate differences in counts between 2 spatial regions. Yeah, you don't want all your data to show up between, you want a real dynamic range to measure on. So the number of genes you get comes up really quickly. And then what you get at the larger spatial domains is the dynamic range you get goes way up.
Okay, thanks. Jeff, can you unmute?
Yes.
Here's Jeff.
Great. Joe, thanks a lot for the talk. I have a few questions. First, you kind of went over cell segmentation briefly. I mean, it's clear how you can see the nucleus. are you just using a density of RNAs to demarcate the cells, or are you using proteins to mark the cell boundaries?
Yeah, we're using integral membrane proteins that exist. It's a cocktail of integral membrane proteins that are all barcoded, and we can image them. So then we end up with a high resolution 3-dimensional image of the cell membrane, the plasma membrane. And that will become the segmentation pattern that we use. So when we measure the XYZ coordinate of an RNA, we will look and see if it's inside that capsule of the plasma membrane, and then we'll know exactly which cell to put it in. So we're not doing any.
Are those for, so you've got a whole stable of them for mouse and for human, so you can do both, I guess.
That's correct. Our human is more advanced than the mouse, but we'll be doing exactly the same thing in mice. That's right.
And what about, like, if you're interested in early embryogenesis? I don't know whether the membranes would show up the same way as they would in an adult.
Yeah, and I mean, there's certain things like even early on, you got to have a sodium, potassium, ATPase, and B2M, and some of these other things in there. But what we did is that custom is an important thing here to these kind of reagents. So I didn't mention it. Antibodies can get custom barcoded. We do it all the time. Abcam is a big antibody company. They can custom their entire IHC catalog, and they have custom little GeoMx barcodes put on them. I think it's about 6,000, I think, is currently what can be done there. Same thing for the RNA. You got unique transcripts that don't exist anywhere else, or unique splice transcripts and things like that. All these panels are made to have custom things spiked into it. And we find that pretty useful when you're really in a situation where the cell surface markers maybe could be very different.
So this gets to my next question, which always has to do with cost, because not only is it difficult to get a grant, but with so much of grants being taken up with salaries these days, the amount that we have for reagents as the consumables is decreasing every day. So what is the cost? Is this going to be affordable? It's beautiful, elegant technology, but it has to be affordable.
Yeah, to give you an idea, I mean, it's if you're going to do whole transcriptomes and space, it costs you about $1,700 a sample. That would be that's all in cost. So that'd be an FFP slide, and that will be roughly 24 whole transcriptomes on an FFP slide. Will cost you about $1,700 all in. So it's actually not that expensive to do that.
And that is on the GeoMx. That isn't the spatial molecular imager. What if we wanted to do an unbiased analysis, single cell, because you were looking at stuff that's never been looked at before, so we want to do discovery.
Yeah, now it's not a pure discovery. I'm not poly-A grabbing or anything like that. So those 1,013 genes we have in there are hybridization probes. That's an in situ, hybridization. Now, nobody knows how these cells are talking to each other, and a lot of things that you'll be measuring, and all these cell types, how they show up in space. We've also done, I didn't show in this, is cell engineering, where we're actually image the guide RNAs, the CRISPR-Cas9 guides, and then phenotypically measure the effect of those guides at the single cell level. But again, it's not pure discovery. It's discovery. Nobody knows exactly the science that's going on there, but it's single cell subcellular at 1,013 transcripts or 100 proteins or whatever.
Well, one final question. This looks to me quite similar to Seqfish Plus or Merfish.
There's a lot of things that are similar. I mean, our, we encoded this 20 nanometer size barcode, which is unique. The other thing you'll find, Merfish covers a large domain of RNA because that's the way it works. These 20 nanometer barcodes give you all that information in one little landing pad. You only need about 50 base pairs of hybridizable material to actually measure that particular transcript, say. So that's sort of different. It's a fully encoded system. It's just not a cyclic system. So when we do proteins, it's pretty much the only fully encoded protein thing. It's not just a cyclic exchanging of colors. They are truly what I call encoded proteins, but it has similarities. If you're looking for something that's the closest to the SMI, it would be MERFISH and SeqFish Plus. You know, MERFISH doesn't have anything for FFP yet, but SeqFish Plus does.
Thanks very much.
I have another question, Joe. So, apart from the whole transcriptome capability of the DSP, is there any advantage of the DSP over the SMI? Because I mean, I can see that
I think it's you can't, at least in my mind, when you actually get into how tissues and organs work, you really have to go to that next spatial dimensions up, because you don't know how these cell aggregates take, and they change their behavior. It's like they really do function as a unit. That's why Mother Nature makes these things as a unit. If you want to understand that biology at that level, I do think you have to measure it at the level that Mother Nature makes function out of it.
But you could, but you could, in the same way that you define regions in the DSP, all the information is there in the SMI to define those same regions, isn't it? It's just that you define things computationally as opposed to using the mirrors.
Yeah, well, if you had, nobody has single cell whole transcriptome imaged at this time, and so you don't get the whole transcriptome in the SMI like you.
Right. Well, so that is a clear advantage of the DSP, is that you can do whole transcriptome. I'm just wondering if
and
there's
the other thing, the throughput, like you're at translational studies, all those Nature papers and Nature medicine papers, you're really running through an entire cohort of samples, 100, 200, 300 samples. The spatial molecular images, all the imagers are much lower throughput than things like a digital spatial profiler. So you actually take a throughput hit, it costs you a lot of time, it costs you significant time. So I really view these things as complementary. I don't really view them as, and trying to figure out the right experiment. I got all sorts of experiments now where I'm doing the private life of these cells that are in like islets of Langerhans or something like that with the SMI. And then I wrap around full tissue, whole transcriptomes. And it's that combination that really matters. We did a lot of that with COVID-19 work, because you got to find the virus, where the virus is, and then you got to understand what the whole transcriptome behavior of the immune context of that virus. And we actually got a recent paper in Nature just on that term. And so I know I'm a big believer that there's not like one Swiss Army knife that gets you all your spatial knowledge.
Right.
Multi-domains are important, I think.
And what will be the upper limit on the number of genes in the SMI?
Yeah, I mean, the way we encode, the encoding scheme works all the way up the whole transcriptome. I don't think it'll be practical. So we'll be launching with 1,000-plex and relatively easy to see, getting to 6,000 relatively easily. These things are all registered in X, Y, and Z coordinates, and they're all super resolution images of those individual 20 nanometer barcodes, so they can get quite dense and you can still resolve them at high sensitivity. And I guess another thing to mention, I guess, the Jeff on those other comparison, the need to have a fully automated system that's reproducible on these biobank samples that don't need any kind of special magic on the upfront, I think is important here. You run into a pathologist. I remember the work I was doing that with the kidney with Evan Newell. It was like they had pathologists that could give us biobank kidney cancer samples out the wazoo, and we could immediately go in there and examine them. And we have sort of accomplished, at least that's what Evan says, and we actually, the same collaboration we had with Dana Farber, is that they accomplished basically 3 or 4 years of single cell RNA-seq experiments, and they'll do it almost in about 3 or 4 experiments that are spatially resolved. The information content, there is no dropouts in these techniques. Like an SMI has no dropouts. Every cell you measure, if it's 500,000 cells, I got 500,000 single cell measurements done. A little bit different than the way.
What is the cost in the SMI? I don't think you said that.
It'll be about the same as we're doing in GeoMx there. It'll be on the order of a couple of thousand or so per sample that you will probe. But that's still early days. We're about, it'll be mid 2022 so we'll be closer to that. We actually are taking, there's technology access programs. I think probably maybe somebody at Sanford Burnham has even done some of those with the GeoMx, where you take your FFP slide, you send it up here to Seattle, you can actually interact with a pathologist, you can get data. We have an SMI TAP program open now, so if you wanted to get some firsthand knowledge of what it's like on samples of your type, you can actually contact us. It's those instruments are really getting booked up, but you can get yourself in the queue for doing some exploratory experiments. In fact, actually, about our first 20 papers that showed up in Nature and Nature Medicine actually came out of people sending us samples through the technology access program and discovering stuff almost immediately, and they published right out of those little TAP studies, we call them.
Right. Yeah, Randy Kaufman here did the TAP study for the GeoMx.
Okay. Yeah, it works pretty good. I think people tend to like it.
Yeah. Okay. If there's no more questions, if everyone has anything, fire it in the chat now. Otherwise, I think we'll wrap up.
Great. Thanks for the invite, Peter. It was fun talking to the Sanford Burnham group, and hope to get down there again someday and visit you guys again. It'd be fun.
Let us know when you're down here. Things are opening up now.
Things are opening up. I used to get to San Diego about 2 or 3 times a month, and probably open up again. I'll get down there.
Okay, great. Thanks, Joe. Thank you. Bye.
See you guys. Good luck on your research.
Thanks.